Hey everybody, welcome to another video. Today we're going to talk about lipids. We're going to talk about the different types of lipids that are going to be involved in terms of human nutrition and metabolism. We're going to talk about their structures, a little bit about their properties, where are they involved in, and classify them a little bit. And in the future videos, we'll talk about chylomicron and lipoprotein metabolism. We'll talk about how lipids are actually digested and how they're going to be absorbed. So there's going to be a lot of videos coming in from lipid metabolism. But first, we got to understand what they are. So let's start classifying them. If you guys like the video, please subscribe, like, and comment down below if you have any questions. I'll do my best to answer. Okay, so when we start talking about lipids, we got to classify them a little bit, okay? So here is lipids, and in this case, dietary lipids that we really care about, okay? What can we classify them into? Well, we're going to have things like fatty acids, okay? We have other things. We have cholesterol, okay? Cholesterol. We have phospholipids, okay? Phospholipids. We have triacylglycerol. Okay, so triacylglycerol. And under this you can put diacyl, monoacylglycerol, whatever. Okay. There's other ones you can put in here. You can put, for example, glycolipids as well, but we're not going to really dive too much into them. These are the main areas that we're going to talk about. Okay. But like I said, do understand that near this topic of phospholipids, we can have things called glycolipids, okay? Glycolipids, where we can add things like, cerebrosides, okay? We can add things like gangliosides and sphingolipids, which again, I'm not really going to get into, but you can find these a lot in the brain, for example, or in the medullary sheaths. But yeah, today we're going to talk about all of these guys, okay? So triacylglycerol, we're going to talk about what it's made up of, what are some of its roles. Fatty acids, we're going to dig into. Fatty acids, by the way, you can actually divide them. You can have saturated fatty acids. You can have unsaturated fatty acids, okay? And you can also have trans fatty acids, okay? We're going to look at the structure of all of these guys and how they kind of differ. Cholesterol is a type of molecule we call sterol. Okay, it's a sterol, really, right? So really, I should actually write sterol, and then underneath it, I should write cholesterol. Okay, and this is really mainly found in animals. Okay, animals. Plants don't make this. Plants actually make other things called phytosterols, and they also make phytostanols. Okay. Phytostanols. And like the most common ones are in human diets are going to be things like cytosterol, as well as cytostanols. There are others, like for example, campesterol and campostanol. Okay. Again, these are the common types of sterols and stanols that are plant derived that you can find in human food, okay? And really, they are very similar in structure to cholesterol, and they actually sometimes inhibit the absorption of cholesterol. So they actually have a cholesterol lowering effect. With phospholipids, these guys can vary so much. And I'm not going to write it here, but we'll talk about it more as we get throughout the video, okay? The thing with triacylglycerols that I do want you to know, though, is that you can abundantly find these guys in your white adipose tissue, okay, or your subcutaneous fat, and they're really just there as a source of like insulation and energy storage, okay? So the main way you actually store energy in your body is through fat, and you really convert them into triacylglycerols, okay? And so they can actually do that. They can also be found in your visceral adipose tissue, okay, where they actually serve as kind of a, mechanical support for your internal organs, kind of like padding, okay? They also have other functions. For example, they carry your fat-soluble vitamins, so your vitamin A D, E, and K. They carry these guys, okay? Hence why they're called fat soluble. This is a fat. And they also give taste to your food, and they can signal for satiety. Again, just multiple functions of these. We're going to look more in depth into the structures of all of these guys, okay? And again, like I said, many different kinds of phospholipids that you can have and many different functions, which again, we're going to talk about, okay? Okay, so let's get started. So here we have a saturated fatty acid right here, drawn out for you in full, and then we're going to switch from this structure into this structure, okay, this way of drawing, and I'll tell you something about it in a second. But I've given you an example of a saturated and an unsaturated fat, and let's go through the difference together. So here's a saturated fat, okay, and you know specifically, this right here is actually going to be stearic acid. This is going to be stearic acid. That's what this guy is called. It has 18 carbons, okay, and it has no double bonds. No double bonds. This is the crazy thing that you have to know about saturated fatty acids. They have no double bonds. No double bonds. Whereas an unsaturated fatty acid, okay, has double bonds. All right? It has been unsaturated or desaturated. That's why it has double bonds. That's where this name comes from. It's from the double bonds. Okay? The double bonds are really just referring to a covalent double bond between two carbons. Either way, this first number is going to indicate the number of carbons you have. So 18 carbons here. Okay, how do I know this? Let's count. Let's count, right? That's not too hard. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and finally 18 carbons. Cool. So that's 18 carbons long. Do you see any double bonds here? No, you don't. Some of you might not know what a double bond looks like. It would just be something like this. Okay, you would have two bonds here. And we'll show that to you in this scenario. Now, there's a couple special regions that I really do want you to know here. I really want you to know these. And it's the fact that this region in blue, this region right here in blue, it is a special region. It's called a carboxylic end, okay? It's a carboxyl group, okay? Carboxyl end. And it's usually just a COOH, okay? But this is how it's actually shown in structure. Okay, so here's a carbon, right? Bound to an oxygen, and, a hydrogen. Well, really, double bounded to an oxygen and a hydroxyl group. But this is sometimes how you would see it written, COOH, okay? But this is a carboxyl group. This group is actually polar. It's polar. You know what that means? That means that this specific region right here, is going to be hydrophilic. So because of this polar end, this region of the fatty acid is hydrophilic, meaning it's soluble in water, okay? But the rest of this hydrocarbon chain, what did I just say? A hydrocarbon chain. Why? Because you have a hydrogen and a carbon, right? So we say it's a hydrocarbon chain, this entire length. The rest of this hydrocarbon chain is hydrophobic, okay? It doesn't like water. It's not water soluble, okay? Now, we'll talk about solubilities and stuff a little bit later, but generally fats are not water soluble, and you guys should know that. Now, so here's this one end. At the other end, though, it has a special group, okay? And this group is called a methyl group, okay? Or we call it the methyl end, okay? Now, this side is actually non-polar, okay? So here's this group. It's non-polar. Here's this group. It is polar, okay? These are the kind of landmarks I want you to remember on this fatty acid, okay? And they're all going to have, at one end, a methyl group, and on the other end, a carboxylic end, okay? So get used to seeing this. Now, the hydrocarbon chain might change a bit, but, the methyl and the carboxyl will remain. So that's what a saturated fatty acid looks like. Now, briefly, we can have different kinds of fatty acids, like different lengths. We can have, 2 to 4, okay, lengths of carbon, which would make them a short chain fatty acid. We can have 6 to 12, which would be a medium chain fatty acid. And we can have even longer. So we can have 14 to, I don't know, 26, and that would be a long chain fatty acid, okay? So it just depends, okay? We have multiple different lengths, and those are going to affect their solubilities, and we'll talk about that, okay? Just introducing you to this topic. So, but that's a saturated fat. And in our bodies, the first saturated fat that we actually make, the first saturated fat that we can actually biosynthesize, the first one is called palmitic acid or palmitate. And then from palmitic acid, which is actually 16 carbons, we actually go to stearic acid. That's the next one we make. Okay, so we start out with palmitic acid in our body, and then we elongate that through special enzymes, and then they turn it into 18 carbon. And then maybe we can introduce double bonds in there if we want. Okay? All right. So, what about unsaturated fatty acids? What's the difference here? Well, actually, there's a big difference. There's a big difference. And you can have something called a cis or a trans configuration of an unsaturated fat. Okay, what do I mean? So first of all, what is this? This is called oleic acid. And notice, how many double bonds do you see here in this entire thing? How many double bonds do you see? Well, if you're saying one, you're correct. Right there. That's one double bond. One double bond right there. So if I were to count the carbons on this guy, okay, I would have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and then finally 18, okay? Each little division point here is a carbon, okay? So I have 18 carbons, and I have one double bond. So how would I write that? I would say I have 18, 1. Okay, cool. Cool. That's awesome. Now, what else? What do you notice about this guy? Is this molecule straight like the saturated fat? Is this molecule straight like the saturated fat? No, it's not. When you have a double bond that is introduced into an unsaturated fatty acid, okay, it produces this sort of kink or a U-shaped kind of configuration, okay? But specifically, this double bond in the cis format results in this scenario. What do I mean by the cis format? Okay, well, where is the double bond? The double bond is right here. That means that the hydrogens in this carbon in here, so I have a CH2 here and I have a CH2 here, okay? The hydrogens are pointing in the same direction. But I can have a trans configuration. What does that mean? Okay? Sorry, not CH2, by the way. This is CH and CH because it's a double bond. But in a trans configuration, the hydrogens are pointing in opposite direction. Look at this. Right here. So there's a carbon here, and there's another carbon there, but the hydrogen, instead of pointing in the same direction, they're actually pointing in opposite directions. But the double bond is still there. Notice how in this trans configuration, okay, the molecule takes a linear form. It takes a linear form, and now it actually looks more like a saturated fat, okay? So it takes on some of the properties of a saturated fat because we have this trans form. Okay, this is important because we're going to change the name of this molecule now due to the fact that it's now trans. We are going to call this guy alidic acid. Alidic acid. So the other one was oleic acid. This one is alidic acid. The only difference between them is the fact that one is a cis form, the other one is a trans form. Okay? So I'm going to put 18-1 cis configuration, and then alidic acid trans. Okay? And it's still 18 carbons, 1 double bond. Still the same thing. Same thing, okay? But the transform turns the molecule linear. Turns molecule linear, whereas the cis form makes a kink, or this U-shaped kind of orientation, okay? Now, since we only have one double bond, and this is an unsaturated fatty acid because there is a double bond, we actually say that this guy right here is actually a mono unsaturated fatty acid, or a MUFA. So mono unsaturated fatty acid, okay? Okay, awesome, awesome. So what else? What else do we need to know about these fatty acids? Okay, well, so the next thing we got to know is how do we start numbering and naming these ones, okay? These unsaturated fats. Because some of them actually have double bonds, okay? And we got to figure out where the double bonds are, but just naming them, 18 carbon, 1 double bond, or 2 double bond doesn't tell us where they are. Okay, well, there's actually 2 systems we use, okay? There is something known as an omega system or a delta system, okay? And some of you may know what I'm going with this. Now, the delta system refers to this. So here's the delta system. Delta system. The delta system starts counting the number of carbons starting from the carboxyl end, okay? So we go that way. So I start going 1, 2, 3, 4, 5, 6, 7, 8, 9. Pause. At the 9th position, my first double bond is present. Okay, remember that. 9th position, first double bond. Let's keep going. 10, 11, 12. Stop. You found your second and last double bond. So, what do you think it's going to be? Well, the first one was at 9th position. The second one was on the 12th position. So you know how we're going to name this? This is actually linoleic acid. And we're going to name it 18 carbons long. Because if you count the whole thing, 13, 14, 15, 16, 17, 18 is 18 carbons long. There is 1, 2 double bonds. Okay. Now, as it relates to where they are, it is at the delta 9 position and the delta 12 position. So delta 9 and delta 12. Why? Because the first double bond we saw was at 9th position. The second double bond we saw was at the 12th position. Okay? So that's how we count from the delta system and how we can write it in the name. There is another system. Okay, and this system is a little bit more layman's terms. It doesn't tell you the position of all of the double bonds. And you'll see what I mean when we start. This one is called the omega system. Okay, so here's the omega system, sometimes also called the N system. Okay, and this is important. Okay, a lot of people will see N and they're like, what the hell is N? But it's synonymous with the omega. And in this system, we actually start counting from the methyl end. So in the delta system, we counted from the carboxyl end. Over here, we start counting from the methyl end. So let's start counting. Here's our first carbon, second carbon, third carbon, fourth carbon, 5 carbon, 6th carbon, stop. We count in this system until we see the first double bond, and then we stop. That's it. So with this system, we can also say that this fat, this linoleic acid, this fatty acid, is actually an omega-6 or an N6. Okay? All right. I'm sure most of you guys heard of an omega-6 fatty acid. This is where that comes from. So here's an omega-6 fatty acid. Okay. All right. Let's get a little bit more practice in. So here we have another fatty acid. Okay. This is an alpha-linolenic acid. Alpha-linolenic. Now, this guy, if you count the whole thing, okay, it's still 18 carbons long. I know it looks a little bit longer. It's still 18 carbons long. So we're going to already put 18 carbons. But what do you notice here? It's got 3 double bonds rather than 2. Okay, so this means that we're going to write 18.3. But we need to start counting. And I want you guys to write it in both scenarios. So I tell you to start from the delta system, okay? The delta system. So we go 1 carbon, 2 carbon, 3, 4, 5, 6, 7, 8, 9. First double bond's at 9. Then we go 10, 11, 12. Second double bond is at 12. Then we go 13, 14, 15. Stop. Third and last double bond is at 15. So how would I write this? 18, 3 double bonds at delta 9, 12, and 15. The other way was from the methyl end, right? The methyl end, that's the omega way, okay, or the N. From there, you start going 1 carbon, 2 carbon, 3 carbon, stop. You met the first double bond, and that's where you stop. So what would this be? This would be an omega-3 or an N3 unsaturated fatty acid. Okay, remember this system is only working for unsaturated fats. Why? Because it was designed to help us figure out where in on this carbon chain is the double bond located. Is it on the 10th carbon, the 9th carbon? Where is it? Okay, and this is how you kind of do it. Okay, if you guys want to get a little bit more practice, I suggest doing this one. Okay, and you know what? Actually, we can do it together very quickly. So if we start from the carboxylin, we got 1, 2, 3, 4, 5, 6, 7, 8, 9 carbon. Okay, that's the first and last double bond. We stop there. Okay, so this one is 18.1 delta 9. Easy. Next, we start here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Same thing. Why does this make sense? It's an 18 carbon long, right? It's 1 double bond, so it's an omega-9 fatty acid, okay? So it's an omega-9 fatty acid. Yeah, remember, guys, we don't only have omega-6 and omega-3, okay? And, you know, this guy will be the exact same thing, by the way. Exact same thing. You counted the same thing. The only difference, again, between this and this is that this one is cis, this one is trans configuration, okay? All right. So, let me point out something to you guys. So what happens if I were to give you the delta number, okay, and I asked you what kind of fatty acid is this? So I didn't give you the structure or anything. I just said, here's linoleic acid, 18 carbon, 2 double bonds at delta 9 and 12. From these information right here, how can I tell whether it's an omega-6 or omega-3 fatty acid? That's what this question is asking you. How? Well, there's actually a pretty easy trick, okay? You take the final number of carbons, okay, which is 18 carbons long, and you subtract it by the last double bond. So 18 minus 12. Okay, what does that give you? What does that give you? That gives you 6, okay? So that gives you 6 carbon, which means this is an omega-6 fatty acid. Okay, why does this work? Why does this work? Because the whole thing is 18 carbons long, and usually the last double bond is listed in the delta system as last, right? So if 12 is here and the entire thing is 18, if I just subtract that, that means that the remaining 6 have to be counted from the other side, from the methyl side. So that's how you know we have an omega-6. So this is a very common question that can be asked, and so an easy way of remembering it is this. Now, you want to know something special about linoleic and alpha-linolenic acid? Something very special. Both of these guys are actually essential fatty acids. These are both essential fatty acids. Okay, hold on. What the hell is an essential fatty acid? That means that we have to actually consume these guys through the diet because we cannot make them. Now, I'll tell you something right now. That is not entirely true. There are metabolic pathways where we can make these, but in such minuscule amounts. So we need to eat these. Now, why are they essential fatty acids. Okay, so let's write that down. Why? And the answer is actually that humans lack enzymes. They lack the enzymes called desaturases. Okay, so the enzymes called desaturases actually introduce double bonds. Okay, they're the ones that actually put a double bond here and here, for example. We actually lack the enzymes that put double bonds on the delta 12 and the delta 15 position. Okay, so this position and that position, we actually lack those enzymes. Okay, so lack desaturases, which again are enzymes that introduce double bonds at the delta 12 and delta 15 positions. Okay? All right. So we actually lack these guys. All right. Now, another thing is that remember last slide I mentioned this guy has one double bond, so he is called a mono unsaturated fatty acid, a MUFA. Well, these guys, they have more than one. They have 1, 2, 3, and 1, 2. What do you call these guys? These are both called polyunsaturated fatty acids. Okay, so PUFAs, polyunsaturated fatty acids. All right. Now, one more thing. What actually happens if you don't consume these essential fatty acids? Okay. Well, you can have a condition occur where you might have retarded growth, dermatitis, kidney problems, and sometimes if you completely abolish these fats, you might actually end up having an early death as well. Okay. So that's why these essential fatty acids are important. Now, with regards to the omega-6 and the omega-3 part, some of you may have heard that an omega-3 fatty acids is actually anti-inflammatory, okay? And an omega-6 is actually pro-inflammatory. Okay, so a lot of people might hear this and they might start going, man, I need to start upping this and lowering this. Not necessarily, okay? In fact, there's not a really clear guideline as to, the amount of which you should be eating, okay? You try to aim for a balance between the two. You don't want to have too much of this or more of this, okay? Because people think inflammation is a bad thing. Acute inflammation is actually very normal and physiological. Chronic inflammation is something that's bad and you don't want, but, acute inflammation is fine. So when you hear pro-inflammatory, don't start freaking out, all right? Just off of this, you can already tell that they exert different metabolic effects, okay? So, for example, one of the key things is that you can make these products called eicosanoids from omega-3 and omega-6 fatty acids, and one of those eicosanoids is, for example, something called thromboxanes, okay, thromboxanes. And these are a class of molecules that are involved in many things, like, for example, vasoconstriction and platelet activation. But the omega-3 version of this thromboxane is actually a platelet inhibitor, okay? So it stops blood clotting. But the omega-6 version of this thromboxane is actually a platelet activator, so it's pro-blood clotting. So you can see how they're already exerting different metabolic effects. And you can find this all over the place, okay? So this guy can actually help with vasodilation. This guy can help with vasoconstriction. Again, different metabolic effects, okay? But again, they're pro-inflammatory and anti-inflammatory, like very different metabolic effects. And you want to aim for a balance between these guys, okay? So when we say omega-3 and omega-6, we're referring to this linoleic and alpha-linolenic. Now, a trick I like to use to remember which one is linolenic and linoleic, because they're pretty similar, and which one's omega-3 and omega-6, is that if I were to split up linolenic, right, you can actually split it up into these three. Okay, so 1, 2, 3 three-letter sections. Whereas with this guy, you can't. So I like to remember it like this. Because you can, split it into three-letter sections, this one's omega-3, and this one you can't, so it has to be omega-6. Now that's just the way I like to remember it. If you guys know another or a better way to remember it, leave it down in the comments. I would love to know. Okay, with that said, let's move on. Let's move on and let's start talking about the different kinds of fats. Okay, so we talked about fatty acids. Okay, now let's start talking about our triglycerides and phospholipids. Okay, so right here, right here is going to be a special kind of reaction I want to start talking to you guys about. Firstly, I have this molecule here, glycerol. Okay, glycerol. Now, glycerol in and of itself is a 3-carbon alcohol. Okay, it's a thick, smooth liquid on its own, but okay, and we can show it right here. Right, it is 1 carbon, 2 carbon, 3 carbon. Okay, it's very simple. Okay, here's glycerol, but then you have these fatty acids. Right, you guys remember the different kind of fatty acids we had? Right, we could have a saturated and unsaturated. Right, so here's fatty acids, and I'm going to abbreviate it as FA. Okay, in this scenario, they're all saturated fats, not unsaturated because there's no double bonds. But it doesn't matter. Right now, what am I showing you? Okay, here's the carboxyl side. This would be the methyl end. Okay, again, carboxyl, and then methyl. Okay, when this carboxyl side interacts with a hydroxyl group on this carbon, they actually form a special bond, okay, a special bond through a condensation reaction. The bond that they form is called an ester bond. So let me show it to you right here. They form this bond right here. It's called an ester bond. Okay, an ester bond is basically just a carbon, oxygen, carbon, oxygen, carbon. Okay, that's how it is. And that's an ester bond. And out of this reaction, you actually release water. Okay, you actually release water. Okay, so you release this guy, and this guy, and they form a water molecule, and you're only left with that 1 oxygen, right? You see that. Okay, and what is this process called? This process is actually called esterification, and it's through a condensation reaction. Okay, now if it's through a condensation reaction, then of course you're releasing water out of this. Okay, now how many waters? Well, you're having 3 fatty acids interact with the 3 carbon backbones of the glycerol molecule, which means you're actually going to end up releasing 3 waters out of this. Okay, so this is plus 3 waters released. What if I want to break this guy down, though? What if I want to break this molecule that I just made, and I'll talk about what it is, down? What if I want to go in the reverse direction? What would that be called? Well, that would be called a hydrolysis. Okay, which means that I'm actually using water to actually break these guys down. Okay, I'm introducing water into the reaction. Okay, now what is this molecule called? This molecule that we just made is called a triacylglycerol. A triacylglycerol. Okay, so tri for the 3 acyls. Okay, so 1, 2, 3. And then the glycerol, obviously being the glycerol backbone, okay? So that's what we're talking about with this triacylglycerol. Sometimes you might see it written as TAG or TG, okay? All right, and that's what triacylglycerol is. Now, something super important about triacylglycerol, by the way, is that this is the main fat that you're going to find in your body, okay? In fact, this is 95% of dietary and stored fat. So this is insane, okay? You got a lot of this triacylglycerol sitting down. And then it's a pretty abundant energy storage form, okay? All right, so what did we learn so far? We had a glycerol molecule, 3-carbon backbone. It'll be esterified to these 3 fatty acids to form a triacylglycerol. Okay, what if it was esterified to 2? What would be called a diacylglycerol? What if it was esterified to 1 fatty acid? It would be called a monoacylglycerol. Okay, same thing, and they're all got this ester bond here. Okay, if you're really curious, an ester bond looks also something like this. Okay, here's an R1 and there's another R right here. Okay, this is kind of another depiction of it, but I think for now it actually suffices for us to know that it's just a carbon, oxygen, and a carbon. Okay, all right, great. That's that. So what's the other molecule that I want to talk to you about? The other molecule that I want to talk to you about, and I'll start with this one because it's a little bit simpler. Okay, and let's actually drag it from here down here, and let's kind of enlarge it a bit. Okay, just so you guys can get the idea. This other molecule is actually going to be called cholesterol. cholesterol. Okay, remember I told you cholesterol is a form of sterol. It's a kind of sterol. All sterols form or have a common property. They have a four-ring core. So here's A, B, C, D, right? 1, 2, 3, 4. This is a four-ring steroid core. They all have this. All sterols share this four-ring core, okay, which is actually called a steroid nucleus. Steroid nucleus, okay? And, it's got a longer name, which I'm not even going to attempt to spell. It's called cyclopentanoperhydrophenanthrene. But I'm sure you guys can understand why it's also called a steroid nucleus. That's just a shorter version of it, okay? I'm not going to even bother to spell that. Now, that's cholesterol, okay? But something about cholesterol very, very interesting. It has this hydroxyl group here, okay? This hydroxyl group poking out here. And a lot of the times when you're seeing it, you're reading the text, you might actually see cholesterol be called a monohydroxy alcohol. And it would be because of the fact that it has this single hydroxyl group, hence monohydroxy there, okay? But, cholesterol is actually very important, okay? It gets a very bad rep in your body. A lot of the times people say, oh, cholesterol's bad or blah, 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 blah. It's actually very important, extremely important for regular, normal physiological function, okay? And, I'm telling you this right now, you don't need to know how to count this, but if you guys are curious, you got 1, 2, 3, 4, 5, then you go 6, 7, 8, 9, 10, 11, 12, 13, 14, you go 15, then 16, 17, 18, 19, and then, you just start going from here, 20, 21, 22, 20, 20, oh my God, 23, 24, 25, 6, and 27, okay? This is how you name cholesterol, okay? Now, you can get this cholesterol from many ways. So you can either get it from your diet, or you can make it endogenously, okay? Endogenously. And I'm telling you right now, the endogenous cholesterol that you make, okay, that cholesterol you make yourself endogenously is actually going to be much higher than the dietary cholesterol that you're consuming, at least for majority of people, okay? In fact, the dietary or the, not dietary, the endogenous cholesterol that you make is about 1 gram per day. It's about 1 gram per day. And, 20% of that, the liver is responsible for 20% of that. And 80% comes from all of the other tissues, with the intestines being the most important. But do realize that the synthesis of cholesterol occurs in every tissue, okay? And we'll talk about the, a watered down version of the synthesis of cholesterol in future videos, okay? We'll talk about certain drugs that can inhibit that. But just remember that endogenous cholesterol, more often than not, is higher than your dietary cholesterol, okay? And in fact, if you reduce dietary cholesterol, your endogenous cholesterol synthesis actually goes up. So again, it's very crazy. And again, I told you cholesterol is only found in animals, okay? Only found in animals. All right. And again, it's very abundant in your brain, your nerve tissues, and extremely necessary. So where the hell can you find this thing? Where can you find this thing? Well, you guys remember from past videos I've mentioned, cholesterol is the prerequisite for making bile acids, okay? It can be incorporated into the cell membrane, which can actually help with membrane fluidity and stuff. If you guys have heard of lipoproteins, it can be incorporated into lipoproteins. Okay, so that these include things like your LDL, your HDL, okay, all of that stuff. And the biggest one, hormones. The biggest one is hormones. Now, some of these hormones are going to include vitamin D3 or cholecalciferol, testosterone, estrogen, even progesterone, some of your adrenal corticoids. Okay, I'll just put corticoids here because I don't have space. But mineral corticoids, adrenal corticoids, all of these different kind of hormones, they are all synthesized from cholesterol. Okay, so you can see how important cholesterol is in your body. It's literally important in so many things. Okay, all right. Now that's all I wanted to talk about with cholesterol. Now, this last thing that I really want to mention is phospholipids. Okay, and I'm sure a lot of you have heard of phospholipids. Okay, this might be something that a lot of people are actually familiar with, but specifically though, phospholipids, they're very different. They're very heterogeneous in their structure. So you have this glycerol backbone again, right here. Here's a glycerol backbone, and there's some fatty acids attached, and then there's a phosphate attached, weirdly enough. So we got 1 carbon, 2 carbon, 3 carbon. Usually the first carbon is called the alpha carbon. The second carbon is called the beta carbon. Okay? Usually, and I mean usually, okay, most often than not, to the first carbon, there is attached a saturated fat. Okay? There is a saturated fat attached to the first carbon, the alpha carbon. To the second one, okay, there is attached an unsaturated fat. And you can see the double bond right here. There's an unsaturated fat attached there. And to the third carbon, this is the important part, to the third carbon, there is attached a phosphate group. A phosphate group right there. Right there. Okay? Why is this important? Why is it important? Well, first of all, we actually call this guy a phosphatidic acid. All right, so this is not a phospholipid at this moment. This is a phosphatidic acid. Okay, sometimes you might see this guy, this group of phosphatidic acids be called glycerophosphatides. Glycerophosphatides, and that's because of the fact that they just have a glycerol backbone and they have a phosphate. Okay. Anyways, okay, what do you know about phospholipids? Phospholipids, they exhibit this thing that's some of you might not have heard of. They are called amphiphilic or amphipathic. Okay, that's the kind of nature that they have. What does this mean? This means that they can actually be attracted to both water and fat. Okay, so they're both water and fat soluble. This puts them in a very, very unique situation, which makes them very ideal structures for compartmentalizing things. As you guys know, right, so let's say here's a cell. Here's the nucleus of that cell. What is the membrane of a cell made out of? It's a lipid bilayer. But specifically, what kind of lipid? A phospholipid bilayer, okay? A phospholipid bilayer. And that phospholipid bilayer is just kind of studded all around the cell membrane, making up the cell membrane, really. And it is because of the fact that it is water soluble and fat soluble, okay? So this unique amphiphilic feature of it allows it to be making up cellular membranes, and not only just for cells. For example, the lipoproteins we talked about, they're actually covered with phospholipids, okay? And I'll show you guys that later. But what else do these guys do? These phospholipids, before we get into their structure a little bit. Well, they can be very useful. They can be very useful. They can be used for synthesis of eicosanoids, okay? And I've already kind of mentioned eicosanoids, by the way. They are lipid-based molecules, okay, that play many roles. They can play roles in innate immunity. They can play roles in hemodynamics. Okay, they can play roles in platelet activation and inactivation, right? So eicosanoids, very, very diverse, okay? Another thing that the phospholipids can do, which is actually very important, by the way, and I'll talk about it, they're good at signaling, being signaling molecules, and they're good at anchoring things, okay? So cell anchorage, okay? And I'll give examples of these, okay? Okay, so let's look at the structure of this, okay? So like I said, glycerol backbone, the first carbon called alpha, second carbon called beta. First carbon usually attached to a saturated fat or esterified to a saturated fat. Second carbon esterified to an unsaturated fat, and the third carbon is esterified to, not esterified, sorry, it's bound to a phosphate. Okay, this phosphate though, this phosphate is what's really completing the rest of this actual molecule. Okay, because this phosphate can bind to polar heads, things like choline can bind, things like serine, things like ethanolamine, okay? Things like inositol, which is actually extremely important, okay? But these heads, these polar heads can bind to this phosphate group to create a phospholipid. And I'll give an example of that right here, okay? These polar heads, one at a time. So we can either be inositol here, or it could be ethanolamine here, or serine here, or choline. Not all of them, only one of them, okay? So this region is going to be the polar head. It's going to be the hydrophilic region that gives it a portion of that water solubility. This region, though, with the fatty acids, this is going to be the hydrophobic portion, okay? Hydrophobic portion, okay? Awesome. And this is usually why we say that the head of the phospholipid is actually going to be water-loving, so hydrophilic, and the tail is actually hydrophobic. Okay, that's why we usually say this. Now, let's take an example here, okay? And let's look at an example of a fatty, not fatty, phospholipid called phosphatidylcholine. Phosphatidylcholine. Note the word choline. Another way, by the way, that we can call phosphatidylcholine is lecithin. It's just another name for it. Okay, lecithin. So phosphatidylcholine. What's special about it? Okay, so let's take a look at it. One carbon here, alpha carbon. Two carbon here, beta carbon, and then third carbon bound to a phosphate. Here's bound to that, unsaturated fat, and here's a saturated fat. What's the difference here? Right over here. This group right here is a choline group. Okay, this is that choline. And it's bound to this guy, phosphatidylcholine, okay? Here's just an example of what a phospholipid could look like, okay? I could substitute this with serine. I could substitute this with ethanolamine. I could substitute it with inositol. And funnily enough, this guy right here, okay? If it's substituted by inositol, for example, okay, or inositol, it would be called phosphatidylinositol, okay? If it was substituted by ethanolamine, it would be called phosphatidylethanolamin, okay? But this guy right here, this phosphatidylinositol, it's very important, for example, for insulin cell signaling. Remember I mentioned cell signaling? It's very important for cell signaling, okay? It makes a specific molecule called phosphoinositol diphosphate, which, if you guys know anything about cell signaling, if you activate a G protein Q pathway, okay, G protein Q pathway, you would actually take this phosphoinositol diphosphate, cleave it, and make secondary messengers like diacylglycerols and inositol triphosphate. Okay, so these guys right here, they're very important, okay, especially this inositol for cell signaling, okay? Now, that's going to be it with the phosphates, okay? Technically, we can have another phospholipid. You might have heard of something called a cardiolipin or a diphosphatidylglycerol. All it is basically two glycerol molecules attached to two of the 4 fatty acids. Okay, so everything is doubled here, and that's called cardiolipin, and it's found in the inner mitochondrial membrane, and it's very, very special in mitochondrial metabolism. It's a type of phospholipid. Okay. Anyways, that's all I'm going to say about that. And one more note really is that just like how we saw that the structure or the composition of the fatty acid can differ in a phospholipid, and this is how it usually is. Okay, the composition of the fatty acids in a triacylglycerol can differ as well. Okay, meaning that not all of them can be saturated or unsaturated. Some of them might be unsaturated. Some of them might be more unsaturated than others. Okay, some of them might be longer length. Okay, you don't know. You just don't know. Okay, it depends on the fatty acids that are attached to the triacylglycerol. Okay, esterified to it. So you just don't know. And this brings me to the point of solubility and melting points. Okay, so you guys have seen most saturated fats are going to be solids, and most unsaturated fats are liquids like oils. Okay, and oils, like I said, a lot of unsaturated fats in them, like a lot of PUFAs and MUFAs, mostly PUFAs. Solids, things like butter, very saturated. Okay, they got a lot of saturated fat in them. But what determines this? What determines this melting point? What determines the solubility? Okay, great question. Great question. So the lower the fatty acid size, okay, and the higher the unsaturation, meaning more double bonds, okay, the higher its solubility, okay, higher the solubility, okay. This actually has a reason. This has a reason. So the lower the size and the higher the unsaturation. Okay, one thing you got to know is that the more double bonds I have, the more polar I am making that molecule, okay? So it makes the molecule more polar. If it's more polar, that means that it's more soluble in water because water is polar. Another thing you have to know with the fatty acid size is you have to go back to the first slide. Remember I said that the almost the entirety of the fatty acid is actually hydrophobic, and only this carboxylic is hydrophilic. If this region was very short, instead of being 18 carbons long or 17 carbons long, it was 4 carbons long. It was a short chain fatty acid. Wouldn't you think that this molecule would be actually more hydrophilic than hydrophobic and more likely to be soluble? Absolutely. Absolutely. Okay? So shorter chain fatty acids, and we'll see this when we talk about digestion. Things like acetate, things like butyrate, propionate, and sometimes even medium chain fatty acids. They don't even require bile for digestion, and they don't actually require to bind to albumin for transport in the bloodstream, right? Because these guys, again, they're very not soluble in water, fatty acids, and they require a transport protein inside of the blood. But the ones that are shorter and have high unsaturations are more soluble. Okay, what else? And by the way, the reverse of this is true. So the higher the size and the lower the unsaturation, meaning more saturated, the lower the solubility. What about the melting point? Well, the melting point, the higher the fatty acid size and the higher the saturation. Okay? Well, then there's a higher melting point. Okay, and this should make sense to you. Again, I always like to think of it like this. Butter, very much saturated, and so it makes sense that it has this high melting point, okay? And if you increase the fatty acid size, it just takes a longer time to, make something that's this many chains long, whereas something that's this many chains long break down, okay? All right, guys, that's going to be everything for this video. Thank you so much for watching. If you like the video, please subscribe. These videos take so long to make, but I hope they help. If you have any questions, let me know, and let me know if you have any suggestions for this. In the next video, we're going to talk about digestion and absorption of lipids. And in the following video after that, we'll talk about lipoprotein metabolism. Okay. Thank you guys for watching, and I'll see you in the next one.